Hello and welcome everyone to PXE International's webinar. Today I'm Christine Vokey, Director of Participant Engagement at PXE International, and the title of our webinar today is Tissue Injury, an Essential Ingredient for Calcification Where You Don't Want It. Today's webinar is a follow-up or a sequel to a webinar that our presenter presented in December called Nature's Stone, How the Body Balances Calcification to Bones While Protecting Soft Tissue. Our speaker today is again Dr. Jonathan Schoenecker. He is Assistant Professor of Orthopedics, Pediatrics, Pathology and Pharmacology at Vanderbilt University in Nashville, Tennessee. He's recently come into the PXE world with his interest in pyrophosphate and calcification of soft tissues, and he's becoming very involved in PXE research. We are taking questions, and Dr. Schoenecker will answer those questions at the end of the presentation. We invite you to write your questions to us anytime. There's on the right side of your screen, there is a question panel, and you can type your question into that panel, and I will be collating them all and asking them to Dr. Schoenecker at the end of the presentation. So with that, I will hand it over to you, Jonathan.
Great.
Thank you for being here.
Thank you very much. Welcome everybody, and thanks for the opportunity to talk with you again today. The goal of this presentation is to continue our discussion as to what is known regarding the ingredients that cause soft tissue calcification. The hope is that if we list out all of the ingredients, we can find the best ways to prevent calcification where you don't want it. Remember, in our last webinar, we discussed the balance of calcium and phosphate and how they are controlled in our bodies. The element calcium and the molecule phosphate are essential for most biological processes. They power our cells and are essential for cell communication. Additionally, calcium and phosphate can combine to make bigger structures, forming big crystals, most notably one called hydroxyapatite, that composes bone. In certain diseases, such as PXE, these crystals are aberrantly deposited into the soft tissues, causing calcification of those soft tissues. Thus, maintaining the balance of calcium and phosphate is absolutely critical for life. And balance is maintained through multi-organ systems, allowing continuous monitoring and balancing of the flow of calcium and phosphate throughout the body. In health, the goal of balance is to provide adequate calcium and phosphate for cell function and to maintain bone integrity. However, remember that to optimize bone and cell function, calcium and phosphate circulate at near saturation, meaning they're ready to form crystal. This is again great for bone, but it's terrible for soft tissues, meaning that the soft tissues should have spontaneous calcification within. As we covered in the last webinar, the body has mechanisms designed to prevent soft tissue calcification. The one mechanism that has become a big focus recently is very important in PXE. This is referred to as the pyrophosphate pump. For more information regarding this pump, please go back and watch our last webinar for all of the little bits and pieces that make up this pump. This pump provides pyrophosphate in the circulation, which enters those soft tissue compartments, which effectively prevents hydroxyapatite formation in those compartments. So, to expand upon our conversation in the last webinar and to help direct future treatments and research to battle these devastating diseases, let's look at the list of ingredients. First and foremost, we have our systems designed to balance calcium and phosphate, namely dietary intake, gut absorption, kidney clearance, hormonal control by the endocrine system. To help balance this, we have the pyrophosphate pump. On the other side of this equation is calcification of soft tissues. In health, as long as we have good diet, good functioning gut, healthy kidneys, a good endocrine system, and a functioning pyrophosphate pump, the balance of the equation favors no soft tissue calcification. We are now learning that imbalances of this equation change the output. For example, the most recent evidence suggests that in diseases such as PXE, in which we have a dysfunctional pyrophosphate pump, the balance of the equation favors soft tissue calcification, leading to calcified soft tissues. The research is exciting as it helps direct potential prevention and possible therapeutics, such as the use of bisphosphonates to replace the missing pyrophosphate. This is changing the balance back in the favor of preventing soft tissue calcification. In general, it would therefore seem that this equation is quite straightforward, and therefore could be applied to diseases such as atherosclerosis, as seen here by this calcified blood vessel, Alzheimer's disease, as seen by these calcific nodules in a patient with neural degeneration, cancer, such as these calcific nodules on a mammogram in a patient with breast cancer, heterotopic ossification, as seen in this patient who has suffered muscle injury, and, of course, diseases such as PXE. The purpose of this discussion is to highlight a very critical element that is often left out of the research and the clinical equation. Specifically, tissue injury appears to be an absolutely essential part of this overall equation. For example, patients with calcific atherosclerosis most commonly have an underlying condition, either genetic, dietary, or environmental, or hypertension, for example, that first causes tissue injury to the blood vessel, and it then forms these calcific nodules. This is known to be the case for Alzheimer's disease, as neural degeneration always precedes calcification. And cancer, which is considered to be an ever-healing wound or a chronic tissue injury, also precedes the calcification within the cancerous tissue. And heterotopic ossification is one of the most clear examples of injury preceding the heterotopic ossification, in that the muscle that turns into bone is first injured or goes under hypoxia, essentially like having a heart attack. This is incredibly important when we consider all of the ways to modify risk in diseases of soft tissue calcification. To highlight the importance of soft tissue injury, I will present to you a very simple experiment that we and others have performed using mice. Specifically, what we do is we alter the diet in these mice in a manner that by this equation should drive soft tissue calcification. Specifically, we provide these mice with a very high phosphate diet, which makes the circulating calcium and phosphate go way above saturation, and again should cause these calcific nodules everywhere. However, surprisingly, when we look at the soft tissues, in particular in this case using a CT in which the muscle, if it has calcification, shows up, what's odd is that in normal mice we don't find any soft tissue calcification. But when we go back to these experiments and consider tissue injury and we modify it, we have a very significant different result. One way to do this in mice is to perform these experiments in a mouse that has Duchenne's muscular dystrophy. The best way to think of these mice is that every time they contract a muscle, they injure that muscle. Essentially, they have continuous skeletal muscle injury. So when we take these mice and we put them on that same diet, as you can see, these mice very rapidly develop calcification of the injured tissues in their body. Importantly, the only soft tissues that become calcified are the muscle, and that's the spot that the Duchenne's muscular dystrophy causes that tissue injury. We have the same results if we just take normal mice and we injure tissues, is we can do it to a point that we drive calcification of that tissue. So going back towards the ingredients, realize that tissue injury is an essential part of these diseases. This therefore suggests that the most common way to develop soft tissue calcification is to have a problem on one side of the equation in combination with a tissue injury. It also suggests that one potential way to prevent soft tissue calcification, even in injured tissue, is to use therapeutics which improve the protective side, such as bisphosphonates. Using that theory, this is exactly what you see. These are the Duchenne's mice without bisphosphonates and a high phosphate diet compared to Duchenne's mice with bisphosphonates and the same high phosphate diet, showing protection of soft tissue calcification despite the continuous muscle injury. Thus, one way to balance the equation is to supplement one side of the equation. Additionally, understanding that tissue injury is an important ingredient allows for the development of even more ways of preventing soft tissue calcification. For example, what if we were to either prevent injury or, as often injury is not preventable, provide ways of improving the healing tissue? We recently conducted these experiments using the same model I presented before and found that if we gave drugs or pharmaceuticals that improve muscle healing, we are able to also prevent soft tissue calcification. Essentially, this means that we are taking the tissue injury out of the equation. So in summary of these two webinars, I propose that the main ingredients that we should focus on in regards to coming up with a comprehensive means of preventing soft tissue calcification in patients with pseudoxanthoma elasticum is to balance diet regarding calcium, phosphate, and potentially magnesium, optimize gut function, assure proper kidney function, assure proper endocrine function, optimize the pyrophosphate pump and supplement it if needed, and to avoid tissue injury. To best understand what I mean by this, realize that tissue injury comes in many, many forms. The most obvious is direct injury. But tissue injury also comes most commonly from chronic diseases, such as diabetes, environmental exposures such as smoking, infection, cancer, and the unfortunate, inevitable aging. These principles are already being used in other diseases of soft tissue calcification such as atherosclerosis, in which we optimize cardiovascular health treating with statins, lowering cholesterol, treating hypertension, all of which function to reduce vascular injury. Same as Alzheimer's disease, in which we provide medications to slow the tissue injury within the brain, and cancer, in which we use surgery, chemotherapy, radiation therapy, etc. to treat the cancer, and in heterotopic ossification, in which we give drugs to try to improve muscle regeneration. This brings up a point that I would like to challenge the other researchers and the community of PXE to consider. That is, why is there a tropism of calcification to the areas of the body that we see in these patients, such as the skin, the eyes, and the blood vessels? The question comes up about this is, what is that tissue injury, and how do we optimize treatments to make it so that we could potentially improve healing of that tissue or prevent injury of that tissue to make it so that we do not develop calcification within that tissue? So again, in summary, I would propose that listing off the ingredients I have here provides a great rationale for developing protocols that shift the balance of this soft tissue calcification equation towards prevention. Finally, and one very important point I would like to make, is that many of the diseases that cause chronic tissue injury we now understand are genetic mutations. Examples of this are genetic problems with management of cholesterols, for example. We know that these patients have increased vascular disease, which predisposes them to atherosclerosis, for example. From this logic, it would only make sense that patients with pseudoxanthoma elasticum who also have a genetic mutation of cholesterol synthesis, for example, would more likely rapidly develop calcific atherosclerosis. To this extent, it may be beneficial to screen patients with pseudoxanthoma elasticum for other diseases known to be associated with chronic tissue injury. So with this and the other webinar, I hope that this presents a global way of looking at how we can manage calcium and phosphate in these patients. And more importantly, from these examples I gave today, is understanding that I do believe we need to come up with the theories as to why patients with PXE have a tropism for calcification within the areas that they do, how we could prevent that, and how we could also potentially give medications to improve the regeneration of this tissue so this does not happen. I really look forward to this discussion, and thank you very much again for this opportunity.
Thank you, Jonathan. That was a wonderful ending to the whole series, and lots more to think about. I want to ask you a question about soft tissue that usually we don't associate with PXE, but I'm hearing it every once in a while, and we have a question about it. One attendee says that she's working with a physical therapist on her painful hips, and she's been told she has adhesions around the hip flexors, and they need to be broken down with a foam roller, which is very painful. And she's wondering if you have any opinions on whether that would be good or bad to actually break those down. I'm assuming they are calcific.
Right. So that is a fantastic question, and the best way to think through this is that tissue injury is in this situation is balanced by how good you are at regenerating that tissue, which is why I presented, for example, in the mice, when we give a medication to improve the muscle regeneration, we're able to prevent further calcification from developing. So the thing is that if you're under a physical therapy protocol in which you are breaking up scar tissue, which essentially was a failure of the tissue regeneration in the first place, and you are able to optimize the healing process after that, we do find that patients tend to get their range of motion back, and they can do well with it. But it absolutely must be the full extent. You have to do everything. You have to make sure that you do the therapy afterwards, make sure you have a good diet during that time to absolutely optimize that tissue of healing back to what it should be. The problem is if you do that physical therapy and break up the adhesions and you don't continue to move the hip, it will come back and come back worse because you added more tissue injury on top of it.
So that brings up the question: how would one optimize the healing process to be able to achieve the goal of not causing more calcification?
At this point, what we do know in our models, and this is all of this I have to say is in mice. I can't jump and say what we're doing in humans. What we have found is that in animal models that tend to develop soft tissue calcification is that if we either limit phosphate or give bisphosphonates at the time of the injury is we can prevent the calcification of occurring during the healing process. In addition, if we give medications or a diet, and the best diet always is a good cardiovascular diet that is low in red meats, etc., is that it optimizes the tissue healing. And then, of course, avoiding smoking, if you have diabetes, having it under control, etc.
Thank you. So you said that optimizing the diet would be one of the best ways to enhance tissue healing. And the precedent then for that, you mentioned it in the context of the balance of calcium and phosphorus. Is the best, you just said now the best is the heart healthy diet. Is that the one that you would recommend?
Absolutely. So we have these animal studies that we do with that, and we're not the only ones. If you look through the literature, there's not a medication that comes close to adding to longevity, for example, than a quote good diet. Most of those diets, what it boils down to is this. Unfortunately, for many of us, eating is actually an injury. And I know that sounds funny, but when you eat red meat, that feeling that you get afterwards, where you feel lethargic, etc., is giving your whole entire body essentially what we call an acute phase response that is much like being in a car accident, going running, etc. Whereas other foods, in particular the more white meats, but then more importantly, more of a vegetarian diet, are known not to cause this. And so what we're learning is that if you're on a heart unhealthy diet, or you smoke, for example, is every time that you eat, you get a bolus of inflammation through your body that essentially causes injury to all of your tissues and really can confuse the healing process.
Hello, Jonathan?
Am I still there?
Now I hear you, yes.
Okay. For my orthopedic patients, for example, we talk about this a lot for elective surgeries in particular, is trying to get to a very good diet, low in red meats. With our mice, we know it makes a huge difference. And I would say that just, for life in general, that's the way to go. It's not as fun, but
Yeah. So, do I understand correctly that you are saying eating at all is an injury, creating an injury?
that's absolutely right. So eating it all is, but the thing is that the injury is incredibly variable. So if you go and eat a burger, for example, and you measure what happens when you go for a great example is you go for a run. Not only do you have endorphins that circulate, but you also have a little of what's called interleukin 6 or IL-6, which circulates in your body. That signifies to your body that you had an injury, being you ran, and you need to optimize blood vessels. And the best thing about exercise is if you do that cyclically, if you do that for an hour a day, as that goes up and comes back down, it promotes your body to stay healthy. The problem is that when you eat or when you smoke, you get a continuous release of that interleukin 6, and it really gets in the way of the normal cycle of tissue uh, being injured and healing. And so the problem is with meat in particular is you get a much, much bigger bolus of that. What's interesting is that if you add certain other things to it, for example, avocado, it actually cuts that reaction down to about half. As I give talks about this, it's been fun. So many people will send me pictures of a big burger with avocado all over it. Uh, but there you really can, and most of you already know this. If you go have a huge steak for the next 2 to 3 hours, you feel awful. Well, what that is the interleukin 6 traveling around your body, and it's equivalent of going for like a 10-mile run.
But you're not getting the benefit of the run.
You are not. It's not directed. And fish and chicken and pork activate that system much less. And if you look on, one of my favorite things is looking on TED Talks. I love those talks. There's a talk on there on how to live to 100, and they really go into this on theirs. Is that they discuss that all of the areas in the world where people live the longest, it's all vegetarian food, all of
Mm.
them.
Mm-hmm. Thank you. We've got a long question from someone. She says you set the challenge to other researchers to answer the question, why is there tropism to this specific area seen in PXE? This seems to be assuming that it is a tissue injury. Is that the only explanation for soft tissue calcification to be triggered, that it's an injury?
No, and that's why I'm bringing this up is that, I come into this whole entire field from a completely different way, and that is that I'm an orthopedic surgeon, and everything that my patients come to me with and everything I do to my patients is a tissue injury. And so my initial interest in soft tissue calcification was that. However, as you get into this field, what you realize is that the body is so well protected when tissue is not injured, even in cases of like, pseudoxanthoma elasticum, for example. And the research that we're about to perform is taking those animals and showing that if you injure their tissue, the rate of soft tissue calcification goes sky high. A great example of this is what the researchers are finding in regards to atherosclerosis in those mice. If you notice, what they tend to have to do to drive atherosclerosis in those mice is give an additional injury. Now, I'm not talking about going and squeezing a blood vessel. What I'm referring to is they put them on a genetic background that's susceptible to atherosclerosis, being the ApoE background, which causes a problem of fatty deposition and injury to the vessel wall. And the parts in which I find very interesting is if you really look at skin, for example, it would appear that the areas that patients get calcification are in the areas of where there's the greatest motion, around the shoulders, the neck, etc. This, what we all have to remember is gravity is an injury. Is that every time that we take a step, we put microfractures in our bones, which actually is good that we remodel them very fast. Same is true with skin. When you stretch skin, it causes an injury, and that process continually remodels. And my best guess is that is probably why those areas have a tropism. The same can be, said in regards to the whiskers on mice, for example, is movement causes chronic injury. Hair follicles are an injury. And so I think that there's many ways that we can test this, but overall my feeling is that that is absolutely part of this equation, and that if we do figure out that part, or we do have medicines that help improve skin healing, for example, is that we will have a way to at least slow the process of the calcification.
Thanks. Someone asked a follow-up question in this vein. She said she developed a painful knot on her knee and went to an orthopedic surgeon, an orthopedist, and was told that she had bursitis, and somehow it had calcified. And this person has PXE. So do you think that that is not normally sort of a problem that we associate with PXE, but this could be because this person has PXE and that was an injury?
Yes. I mean, the research we're about to do, as soon as we we've been waiting to get a hold of the mice from the Weedo group. If they're on here, I can tease them a little bit. Their mice have mites, and so their mice are dirty. And so we're ready to do, the orthopedic lab here we have set up is we do all of these tissue injuries from every type of musculoskeletal tissue. And that's the exciting part of what we're about to do is we're going to take all of the principles that we've done before. For example, we're going to take these mice and put them on the Duchenne's background. And our best guess is what we're going to see is that any musculoskeletal tissue that we injure will end up developing at least microcalcification, if not complete HO, like what you see in the mice that I showed here.
This question sounds way out there, but I'm going to ask it. Is there any connection between PXE calcification and having problems or trouble losing weight?
PXE calcifications and losing weight. Can you be a little more specific?
She just says her metabolism is 4% more efficient than the average American, but she's unable to lose weight, and she eats a completely vegetarian diet. Just wondering if there's any connection that you could see.
Not right off the bat. I would have to really think about that one. The question would be whether or not she also has an endocrine dysfunction. And I bring that up because we often, with the screen I have up there, we often try to jump to the tree and not look at the forest. What I would suggest to anybody who is doing all of the right things in terms of diet, exercise, etc., who can't lose weight, is to have their endocrine system evaluated. I can tell you from a personal experience, not in terms of necessarily weight, but my wife went through something similar, and right away we found it, fixed it, and everything healed up. That's why I really think when you look at what I have up here on the screen, is there are really easy ways to evaluate all of the things I have up there, especially endocrine, kidney, gut, and diet. And that's what I would suggest, especially if you're having a hard time losing weight when you don't think you should.
That's a great answer, and gives a credence to the idea of the the whole system being important here. We have another question about kidney stones. Since gut and kidney are key in the formation of calcium oxalate kidney stones, due to the failure of oxalate and calcium to bind in the gut and instead binding in the calcium forming the stones, is there any correlation between this and gut kidney portions of soft tissue calcification?
In regards to the kidney stones related to other soft tissue calcifications?
Yeah, and particularly could it be caused or enhanced by PXE that forms?
I would think so. I can't say whether or not that has been tested yet. The thing is that what I want to highlight here is this. I spent years studying calcification. And what I'll tell you is that, and this is true in the PXE community in terms of the research, the output that you get in these mice, the microcalcifications, they're difficult to measure. They're difficult to quantify. One of the best advice that I ever had when I was doing my graduate work was anytime you study anything, always try to study it more out of control. And when you say more out of control, really what it comes down to is adding more tissue injury. So in order to answer these questions, what I would propose to anybody studying these is to take the model that you're studying and make it more out of control. A good example of that is what we did with the Duchenne's. Originally, we were just crushing muscle, and we saw microcalcification. But in order to increase the signal in what we saw, we went to the Duchenne's model. Similar is true with all of these other diseases, such as the vascular disease in PXE, is again, researchers have done really well by adding the ApoE on top of it. The same would be true at looking at calcification within the renal aspect of this for calcium phosphate depositions or calcium oxalate within kidney is to make sure that that's studied on a model that doesn't necessarily give the stones but puts it just to the point of going over the top to see if a PXE mutation would end up being the thing that would push it over. I think that's going to be the main way we'll be able to figure that out fast. Otherwise, we just have to study the natural history in patients.
I'm glad you mentioned that because I'm going to give a big push for filling out our new survey at the end of your presentation, at the end of this, the questions here. That's where we're going to learn all about the natural history and about all the different things and conditions that people with PXE have. I've heard of people saying my muscle, some doctor told me my muscles are like cement. That isn't something we hear all the time, but
right.
In the context of what you're saying, it could very well be due to the fact, is this correct, that they have PXE and had an injury?
Yes, I am. I believe the problem is when most people look for heterotopic ossification, they're looking for extreme cases like I showed. However, just like the reason why we do mammograms is that most of the heterotopic ossification is actually microcalcification. And so the thing is that a typical X-ray more likely would not pick up that necessarily. There have to be a little bit more specific tools used to look for it. But we should be able to answer this pretty soon as we're able to get rid of the mites from the mice.
Speaking of the microcalcifications, you had a slide of a mammogram of someone with cancer who had microcalcifications in her breast. And we have studied that in PXE and found that most women with PXE have those microcalcifications, and for the most part, they're not cancer.
Right.
So that's just to let people listening to the webinar know it's not.
That's important, and I want to make a point about that that's very important to understand is like what I have listed on this slide. Tissue injury comes in all forms. And so the thing is, again, just living is a tissue injury. And so I use the cancer as another way of saying tissue injury out of control like we do with our mice. But the thing is that all of your tissue, including the interstitial tissue of breast, is injured all the time. And so the thing is that what that more than likely represents is just continuous injury to the normal tissue that's there. Cancer puts it over the top because it's a continuous injury. Yeah. But I agree with you as I want to make sure that that is emphasized here. Just because you have calcification within breast tissue does not mean that you have cancer. it means that there likely is an injury in that area, but it doesn't have to be from cancer.
Mm-hmm. And again, maybe PXE is somehow providing that injury process. Men's testicles, pretty much across the board, according to our small study, have microcalcifications as well, and it's not testicular cancer.
Right.
One more question about diet. You mentioned meat, and it's still, it's injurious effect. What about dairy?
In terms of the amount of calcium and phosphate that you get from it?
Or you were saying red meat in particular gives
Oh,
you.
I see. Great question. So what I can tell you about dairy is it definitely does not have the same bolus of the IL-6 that's produced that meat does. There are two reasons, and we don't know which one it is, as to why meat in particular is worse. One of the first is the easiest, except for it hasn't been proven, and that is that the greatest source in your body of IL-6 is muscle. It's a way that the body communicates to the rest of the body that you need more oxygen. So what happens is when you go get a good workout and you quote feel the burn, you just gave your entire body a huge bolus of interleukin 6 or IL-6. What it effectively does is go to your liver and release all kinds of wonderful factors that are important for tissue regeneration and to promote new blood vessels. And so the thing is that that's how you get in shape, is that system. Mice that don't have IL-6 have a hard time getting in shape. And so the thing is that if you eat meat, what we don't know is that if the IL-6 from the meat that you ate is what's giving you that bolus, or whether or not the actual construction of the muscle itself within your gut is injuring your gut, and your gut muscle is releasing the IL-6. That is not known. That's the main reason so far that we know as to why a meat diet seems to cause more of an inflammatory response than other foods.
Mm-hmm. I think that's all of our questions. Someone commented that it's been really great to get a different sense of what is meant by injury. You've really expanded our outlook on that. We really appreciate the insight you give, the new insight you give to PXE and PXE research. I am going to put up a final slide if there's no more questions. We have a 20th anniversary meeting coming up. It's in October, and since I can't see the slide, I can't exactly tell the date. But I would like you to think about coming and joining us. It will be partially a research meeting as well, with researchers there to tell you about the latest knowledge on all the things we're learning with PXE. This webinar and Jonathan's previous webinar are already the previous webinar is already up to view, and Terry sent everyone in the audience a link to view the previous webinar, and this one will probably be up to view within a week, so you can see that from our website. And I wanted to plug the PXE help survey. You can start it right on the homepage of our website. It is a very extensive questionnaire asking you about your PXE and all about the rest of your health too. We're trying to get a bigger picture of all the manifestations that people with PXE have to find out, just anything we can find out. Everything you can tell us will be important there in understanding the natural history of the disease and helping us be ready for clinical trials of treatments that it sounds like are not too far away. So, Dr. Schoenecker, with that, I want to thank you for, again, presenting with us. You are very understandable, a great teacher, and I've enjoyed listening to your presentation. So thank you for being with us, and thank you all the guests for your great questions and for being with us as well.
Thank you for the opportunity.
You're welcome. With that, I will end the webinar. Thank you, everyone, for coming.